It was Food Safety Week last week, so we're sort of carrying on with the concept of controlling aflatoxin to improve food safety. So, we're just giving a bit of an outline what's aflatoxin, the incidence, impacts and regulation. I'll be talking about some of the work we've done in maize in Australia and maize in Africa, and then Arsen will follow on with his work in peanuts. So it's all an aflatoxin sort of concept and the crops it affects. So aflatoxins are mycotoxins, toxins produced by fungi. So mycotoxins are toxic metabolites or secondary compounds produced by the molds and the fungi on almost all agricultural commodities worldwide. They're natural toxins. They occur under natural conditions in animal feed as well as in human food. Fortunately, not so much in Australia. Economic losses occur at all levels of food and feed production, including crops and animal production, processing and distribution. Some of the mycotoxins, particularly aflatoxin, are regulated, so there's an economic impacts. It's estimated that 25% of the world's crops are affected by mycotoxins. They're often invisible, tasteless, chemically stable, and resistant to temperature, storage, and most processing. They can affect at any stage in crop development, from when the crop's in the field, pre-harvest, during harvest, silage, storage, and along the value chain. Basically, at any point where the conditions are favourable for fungal growth. There's lots of different mycotoxins, basically more than 400 that are known. They come under a series of different classes. The 6 main ones are listed up there: the aflatoxins, trichothecenes, fumonisins, zearalenone, ochratoxins, and ergodalkaloids. These cover a wide variety of different chemical classes. And I'm a chemist, so I've drawn some nice chemical structures up there for just showing the diversity of chemicals we're talking about. The ones we're talking about today are the aflatoxins. So, aflatoxins are produced by Aspergillus fungus, particularly Aspergillus flavus or Aspergillus parasiticus. The main aflatoxins are shown there are B1, B2, G1 and G2. It's really good naming. The B1s fluoresce blue and the G1s fluoresce green, so it's sort of pretty basic naming, but there's similar structures. Aflatoxin B1 is certainly the most common one, and it's actually a class 1 human carcinogen, so it's strictly controlled in a lot of products. It causes liver disease, it's hepatotoxic, carcinogenic, tetragenic, causes hemorrhages in the intestinal tract and kidneys, reduced growth rate, also known as stunting. A lot of Africans that I came across were actually shorter than what they should be because of aflatoxin exposure as young children. Immune suppression. If in some of the countries where there's HIV, the compounding effect of aflatoxin is quite impressive. Poor performance of livestock. So I belong to the Centre of Animal Science, so it actually does impact on animal health. In Australia, it's more an animal concern, certainly in maize. In Africa, where we've done some work, it's a human impact. So Aspergillus in the maize field. Aspergillus flavus it's a ubiquitous fungi. It's present just about everywhere. It only becomes a problem when it actually infects the product. So it's in the soil, you have crop residues sitting around from the previous crop. It then transfers down the silks. Most people are familiar with sweet corn, with the silks that come out of the top of the cobs. That's how the fungus gets into the cobs, transfers down the silks into each of the kernels. So it can infect the crop in the field. It can actually infect post-harvest during storage. Even if you kill the fungi, the toxin that's already been produced is still there. As I said, it's a worldwide problem. It's majorly of concern in the tropical, subtropical climates. It's on the global map there, they've drawn the 35 degrees north and south, so the band within there is certainly the band that's most affected. It can actually go slightly above and below. Stress factors that weaken the plant's natural defences and promote fungal colonisation include things like drought, poor fertilisation, high crop densities, insect or mechanical damage. a lot of the things that we try to control in the more developed countries, but not necessarily as well controlled elsewhere. It becomes quite a complex system trying to control it. So they have these wonderful little pretty diagrams that show all the overlapping spheres of things that you're trying to control: the breeding, the crop management, storage, sampling and analysis, the impacts of regulation. It's really an integrated approach to solving the problem, and all parts of it become important. So our previous exposure in Australia, we had a large mycotoxins in maize GRDC project in 2003 to 2009 with a student who did surveys of the Australian maize production. There is aflatoxin there at low frequency and concentrations. Fumonisins, one of the other mycotoxins, is more common, lesser occurrence of some of the other mycotoxins. They developed a management plan because there had been major impacts of mycotoxins where shipments had been rejected from overseas. So the industry as a whole wanted to make a concerted effort. So they came up with these rather simple looking statements. They're 5 prongs of communication and coordination across the industry: prediction and prevention, rapid detection and assessment, effective use of contaminated grains. That in Australia we have different regulations so that we can actually the grain that's actually contaminated with aflatoxin, as long as it's below a certain level, can be reused as for livestock. So it gives us a second use for things that have been contaminated. And breeding is an obvious one. So this is maize in Australia. Its total production, it's a rather minor crop, 450,000 tonnes per annum. It produced from tropical Queensland down to Victoria. It's a well-regulated industry, large-scale production, mechanical harvesting. The grain is tested on receival. They have testing programs where they test multiple probes across a shipment load to measure the amount of aflatoxin. Aflatoxins, it's not uniformly distributed. You'll get hotspots. So they need to be able to test the whole shipment to actually assess how much aflatoxin's there. And then the clean controlled storage, we have, nice grain is being produced. So in Australia, aflatoxins do affect the grain crops, including maize, but generally not a major health problem. It becomes mainly on trade. It's an economic impact because there's regulatory limits placed on the permitted levels of aflatoxin in different commodities. If you look at that thing there, in human food, we only allow 5 parts per billion. So if that shipment of our grain comes out to be higher than 5 parts per billion, it might be downgraded as to the 10 parts per billion or to the stock feed for dairy cattle or the stock feed for cattle. So you can understand, if your grain gets downgraded, you get less dollar return. So that's the economic impact. So there were serious domestic problems in Australian maize back in 2003, which is why we had the GRDC product. But there's been occasional export rejections even since then of whole shipments of grain. Every country overseas will be testing things like maize on arrival. So they'll test the shipment. If it fails their regulation, they will turn it away and not pay you the money. As you can understand, rejecting a whole shipload of grain is a serious economic impact. So the grain industry in Australia is very much focused on keeping the levels down. So generally in the lower aflatoxin risk in developed countries, because we have improved standards of living and agricultural practice. So not only is the levels lower in the crop, but because of our living standards, we actually eat a very diverse diet. So you're not eating grain day after day after day. But in Eastern Africa, maize is consumed as a dietary staple, and the aflatoxin contamination poses a significant threat to food and nutritional security. So they have chronic exposure, causing the cancer and stunting in children, and the acute exposure causes death, with a number of outbreaks reported, which is, it's an interesting term, but they don't just term them as outbreaks. People die. So that's where Kenya is in Africa, where we've been doing some work. The area is mainly on the eastern part of Kenya where they grow maize. They've got, these are some of the levels that they reported in maize that was being consumed by people. So our regulation is 5 parts per billion, so you can imagine 8,000 is not a good number. Even as recently as 2010, there was 2.3 million bags of bad maize that was impounded. Unfortunately, it then disappeared, so no one actually knows what happens to it. But the suspicion is that it actually entered the food chain. So yes, in Africa, aflatoxin actually makes the paper, the common daily newspaper. People know about it because it's just seen as a major impact on their lives. So in the past few years, we've had this AusAID project, Capacity and Action for Aflatoxin Reduction in Eastern Africa, or CAAREA. So in this, we worked with a large team of people. You can see all the diverse affiliations, including the UQ one, so that's myself, and Glenn Fox is hiding in the background there. But it's been a very interesting time seeing a different side of aflatoxin than what we see here in Australia. So in Kenya, maize is the main staple for 96% of the population. 70 to 80% of maize is produced by small-scale subsistence farming. You can see the amount that's consumed exceeds the domestic production, and part of that is because the typical yield is only 1.5 to 2.6 tonnes per hectare. On their research stations, they can get up to around the 5 and 8 tonne, so they can clearly see why they have a deficiency of food. They have a high incidence of pest and disease and the mycotoxinogenic fungi. That's just some photos of the small-scale production. Each of those sort of terraces and could actually be a separate farm. Harvesting and drying occurs on a rather primitive level, and rudimentary storage. Each village will have its own mill, so they're milling. So each farmer's producing his own grain, they're milling it, and they're consuming it on a family basis. So your maize may not look quite as good as what the maize we get here. Within that system, there is little or no capacity for testing because we're not talking about a large scale production. The economics of it's almost against it. In the Kenyan cereal mills, they do make maize flour to be sold, but then they have these trucks at receival where the trucks will actually contain multiple bags from multiple sources. You might have one contaminated bag in there, and it becomes a logistic statistical exercise as to how representative any sample they take will be. And of course, not all mills test for aflatoxin. So this is what our project was focused on. The part that I was largely involved with was the establishment of the diagnostic platform at Becca at ILRI in Nairobi. They had other parts where that identifying resistant maize, germplasms, and modelling in the field. So in Kenya, they do have an aflatoxin limit. It's set in the regulations. It was 10. I think it's now actually gone down to 5. But it's only of meaning if people actually test for it. Interestingly, dog food apparently is tested. So our project set out to establish an analytical chemistry lab to analyse the maize samples from the project field trials and the farm surveys. So we were literally thousands of samples. That's some photos of the lab, the capacity we have developed over there from the people with the... It's not only the infrastructure we developed, it was their safety standards, their ability to deal with the number of samples, the data management, the lab management side of it. So we don't have any photos of the before because basically it was a bench and one person, James was the only one who was there at that stage. So coming out of the project, we have put a fair bit of infrastructure in there, and they are quite well equipped to use it. In fact, one of our students is currently over there working in that lab. So the sort of techniques we had for analysing aflatoxin range from the strip tests through the ELISAs, the HPLCs and LCMS. In the recent months, we've actually just put on LCMS. We had a HPLC at Becca, and we've now actually attached the mass spec side of it. So we've gone from a lab where we had molecular biologists trying to learn chemistry. We actually have real chemists employed, and they're actually running LCMS. As part of the project, we did inoculated field trials to try and test the resistance of the maize. and determine which of the maize strains that they currently use are actually more resistant. So in previous times, they actually only looked at the actual production from their maize, so which maize was actually more productive in an area. There was no selection based on aflatoxin because nobody could test for it, so therefore you couldn't select for it. So we did trials across four sites, up to 7 reps, 10 inbred lines, and we analysed literally thousands of samples until you get plots that look like that across the different entries at the different sites. I won't bore you with the stats on those. But there was strong evidence of the G by E effects, and the modelling actually suggests there was a fair bit of management factors. So out of that, the research institutes were selecting which maize to recommend to the farmers. So the ultimate aim of the AusAID project was to make maize safer for people in Africa, but a secondary aim, because it's AusAID, is capacity building to provide both the research infrastructure, which I've shown you pictures of, and also the scientific training. So we've had AusAID higher degree students both here at QAAFI. Titlea is sitting here as one, and Ben's currently over in Becca. And we've also had Vincent, who did a master's here as well. So I'm just going to give you a brief overview of some of the projects the students have been working on. So the single kernel analysis, we've devised ways of actually analysing on a per kernel basis just to show the distribution of aflatoxin through a sample. And there's extremely skewed distribution. These samples down here are typically about 150 kernels per each. The average aflatoxin in sample 1, for example, was 668. 94% of the kernels were below 20 parts per billion. But some of the kernels had levels up to 85,000. And in the second sample, there were 4 kernels, 35,000 to 55,000. So those individual kernels were the problem, not the majority of the sample. So just doing a small amount of analysis on the statistics, if you remove the one kernel at the 85,000, the average would drop from the 668 to 100 parts per billion. If you took the 4 kernels that are above 1,000, you could get it down to the remaining 146 kernels down to 6 parts per billion. So basically, if you could actually sort those kernels, you can make a bad sample good. Something that was completely unfit for consumption now becomes well below our 10 parts per billion that we were looking at. So we were looking at NIR sorting, doing correlations, using the NIR absorption measured in grain and correlating with the analytical methods. Unfortunately, the aflatoxin levels are significantly orders of magnitude lower than the grain constituents. To date, some of the best data that Titlea has managed to get is the PLS regression coefficients in the germ down kernels is only 56% orientation. There is a trend there, but it's being masked by the other chemical constituents that are in the samples. We have had access to a kernel sorter in the labs at Becca, and you can sort the kernels, but there's a fair bit of positive, false positive rejections, and they're generally considered to be too high. It does show promise, however, and so Titlea is continuing with her work looking at the metabolomics to get a better understanding of the chemical changes associated with the aflatoxin accumulation. So the hope from this is that we can actually look at other chemicals in the grain that are changing, so that we can get that better correlation and be able to sort our grain. Photodynamic inactivation. The death of microorganisms upon exposure to dyes and visible light was first reported over 100 years ago, but the concept's principally been used in fields such as cancer treatment. In recent years, photodynamic degradation has been shown to be effective against food microbes. So we've been investigating the application of this to controlling the Aspergillus fungi. And Yasmina has actually had been doing a lot of work in her lab looking at this. So it works by killing the microbial cells through the photosensitizing light-induced cytotoxic reactions. So the light excites the dye, or we've actually been using some natural plant extracts that actually do this as well. So they get excited to the triplet state. and you end up getting the cell death. So you can actually kill the fungi cells in the sample. So Ben's been working with curcumin as some of his initial trials. So he did firstly in vitro, looking at different concentrations of curcumin and light intensity. And that's some of his results. He's achieving 3 log drops in CFU by irradiating at 420 nanometers. So we're talking about visible light. And curcumin concentrations, the optimum concentrations are in this range, the 15 to 50 millimeters, 50 micromolar. He's also then moved on and tried in vitro experiments where he's applied the photosensitization to both whole kernels and mill kernels. The drops aren't quite as impressive, but we're still looking at 2 magnitudes of log drop, which for a non-osmosensitive system is actually showing considerable potential. We actually hope that we can actually put this in some of the cereal mills because they actually wet their grain before milling. So the concept of being able to wet it with something that contains small amounts of curcumin and irradiating it does have potential in a practical application. Another part that we have been looking at recently is bio-absorbents. Because there's so much mycotoxin in the feed, people use bio-absorbents such as clays, charcoals, aluminosilates, specific polymers, and most recently bio-waste such as grape pomace. In Kenya, they're actually talking about using adding clays to human food in times of when the aflatoxin levels are so high. Such a thing would not be considered in Australia in human food. So we've had a student recently who was looking at bio-waste as a banana peel. The student's actually not an African student, but the project is equally applicable to the African scenario. Dried banana peels previously been shown to be effective as a bioabsorbents for things from dyes, heavy metals, pesticides, and clean up of wastewater. So the question was asked, could we use it for aflatoxin? So Zahid has done some studies in our lab in his 6 months he spent with us, looking at the percent absorption under a number of systems, but this is just his results over pH. So the percent absorption can be up to 80 and 90% absorption of aflatoxin in a solution at pHs that are reasonably within the physiological range. And it's a rapid absorption. So we're talking within a few 10 to 15 minutes, getting absorptions up to 80% of aflatoxin. So we're hoping to take that forward as something that actually it's a low-cost decontamination method. So that's a summary of the work we've done in CAAREA. It's been perceived as a highly successful project. It's now been funded by DFAT into a further phase from 2014 to 18 for the odd 4.4 million. Unfortunately, I don't get to see much of that, but it's nice to be part of such a large project. And if anyone knows, the amount of DFAT funding was cut back severely with the previous government. So the fact that we're getting that sort of money means we're actually having impact. So the higher degree students are continuing with their work from Tidlaya, Flad, Ben and Zahid, who's in Pakistan but is busily writing papers for me.